Hello again, and welcome to part 1 of chapter 11, which is the nervous system, part 2, getting more in-depth with the nervous system. And you may have noticed there's this distinct time jump between, and cat jump that's happening right next to me at the moment, between my two boys here. And what happened was I recorded this, and the recording is fine, except for OBS, for whatever reason, decided not to pick up what I was doing in the background on my desktop, so it just showed literally this screen you're seeing right now underneath me, and that's it. So I'm talking away, giving my lecture, and you don't see any of the screens, any of that, nothing. And I guess I could have gone back in and edited it, but in a way it's a little faster for me to listen to the cat scream in the background. See, this is why I don't record in the late evenings. The kitties are all awake and they want to play and complain and talk about things. One of them is looking and the other one is looking at me. But me and Monty are awake, and this one was kind of short, so I decided to re-record this. So, welcome back to almost 10 o'clock at night, which is a whole 12 hours removed from when I first recorded this sucker. Fun! So anyway, let's get into it. It's all good. So, now that we talked about neuron transmission and how all that works, let's start getting into the nervous system. So we're going to be talking about the central nervous system first, and then we're going to be breaking it down from there. So let's talk about your brain and your spinal cord, because this is kind of important because these guys control everything in your body. So in your brain, the outer layer is gray matter, whereas the white matter is located deeper in, and we do have some islands of gray matter located throughout. If I was in my lab, I actually do have a human brain in a jar, because how could I not be a mad scientist without a human brain in a jar? And however, in your spinal cord, this is reversed. So there's white matter on the outside and more gray matter on the inner cord. Now, there's a reason for this. It's because, remember, the myelinated sheaths, which make up the white matter, make transmission of signals up the nerves of the tracts go way, way faster. And we need that, especially when we're in the middle of stepping on a Lego, and we need fast reaction times to let us know, get your foot off the Lego. It hurts. So that's why on the outside you've got of your spinal cord it's white, and on the inside it's gray. So the brain is attached to the spinal cord via the brainstem, with axons bundled in ascending tracks for signals to go up to the brain, and then descending tracks for signals to go down through the spinal cord. We'll be looking at that later. And both are connected to the peripheral nervous system via the peripheral nerves. And the peripheral nerves are actually kind of interesting, because if you can actually tease out the entirety of a nervous system, it looks kind of interesting. And some doctors have made names for themselves from doing a dissection like that, where you pull out the whole nervous system. And some doctors have, and especially in the past, have gotten in trouble for that. So nervous system dissection. Let's see if I can find it. Not diseases. Yeah, dissection. Yeah, here's what you'd look like as just a nervous system. So there are some very famous ones that we've pulled out completely. Like, for instance, the mystery of Harriet Cole is one of them. Yeah, you can find this in, oh gosh, yeah, so this is interesting. We're not entirely sure what the full story is. So after she died and willed her body to the Hannerman Medical College, they stated to refer to Harriet that way. She had been cleaning woman at the school until her death in 1888. And then supposedly she left her body to be left to science, which was okay, maybe. And But the school's most famous anatomy specialist, Professor Rufus Benjamin Weaver, got his hands on the body and it became basically extracted it and mounted it, and you can still go there and see it today. It's a part of, I believe, let's see here. So, yeah. Experts told him it was impossible, and he did it. And actually, back then, like I said, this was quite a feat. You can actually, people made their names doing this. Yeah, so there's Harriet Cole. You can actually go see her. What? Yeah, okay, I'll have cookies, whatever. So anyway, and it's basically at Drexel University now. And here is Harriet. It's at Drexel's Queen Lane campus. Other places that could have been other things right here. So, it's kind of an interesting story. I mean, it is an amazing work that he did. And like I said, very few at that time could pull off that level of teasing out somebody's nervous system. So, good on him, I guess. But at the same time, now everybody's staring at your naked nervous system. It's kind of weird. So, anyway, do we know what the true story is about how her body was given to it? It said she was a black woman who worked as a maid or a scrub woman in the university laboratory, died in the late 1800s, and donated her body to the medical school. And then the story goes, was dissected by Weaver, preserved and mounted as a teaching tool, and as a masterpiece of medical specimen preparation. But it kind of is like there's a lot of mythology around it, and we're not sure if that's truly Harriet or if she gave her body willingly. All of this is kind of like, So, yeah. So, finding the truth of Dear Harriet has been kind of a major thing of speculation, especially since, this is the late 1800s, Not everybody had rights, and hopefully you know where I'm going with that. So it's a fascinating thing if Harriet Cole is the actual person that's named in the nervous system, or that she had any decisions regarding what happened to her after her death. If you kind of get where I'm going here. So it's kind of interesting. So, and again, and this article goes over it. And so if you ask, like, did she donate her body? We don't know. So Harriet Cole could, and her nervous system could definitely be not willingly. So that was kind of interesting. Anyway, so I wanted to point that out. Like I said, teasing entire human nervous system out is insanely crazy. Here's another dissection that was done. This one is like psycho old. I believe this one was pulled out. Yeah. This was a pair of medical students took 1,500 hours dissecting this nervous system in 1925. So, and this was done in Missouri. Like I said, to be able to do this is, yeah, that takes, trust me, I had to hack the brain out of a shark once. Yeah, that took forever. So I don't even know how you'd have the patience of a saint and all sorts of insanity. So yeah, like I said, we have some interesting intact human nervous systems. Like I said, some of them come from questionable sources. Anyway, where we were going with that, just to let you know. All right, let's go ahead and get back to the brain, because, we got to talk about the brain. So the meninges are the membranes that are found in between the bone and the soft tissues of the nervous system, and they protect the brain and the spinal cord. So there are 3 layers to the meninges. You got your dura mater, which is this tough white connective tissue that contains many blood vessels and nerves, which includes the dural sinuses. where the dura mater splits into two layers, forming channels through which blood flows back to the heart from the brain. We have a brain-blood barrier, and we're going to talk about that in a minute. And basically what that is that we want the brain, the blood to come up and nourish the brain, obviously, because we need oxygen to the brain and we need glucose, because like I'm going to tell you in a minute, your brain's a hungry boy. About 25% of everything you eat goes to your brain. So this is why we get hangry. But at the same time, there's things in the blood that we do not want to carry back over into the brain. It's a way to protect the brain and a way not to get feedback when we're putting out things like hormones and whatnot, which is what we're going to be talking about in Chapter 13 when you start Anatomy and Physiology 2, or the BIO169. So we start right back in with the endocrine, with the hormones and whatnot, and talking about that. So the arachnoid matter is the next one. This is thin web-like membrane without blood vessels, and this guy is part of something that starts with a fluid that goes around and in your brain called the subarachnoid space, which is between the arachnoid mater and the pia mater, where the cerebrospinal fluid, or the CSF, is found. And this acts like shock absorbers for the brain. So that way we can shake our head and our brain doesn't go ow, 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 ow. Because we don't want it flopping around in there. So we have the cerebrospinal fluid that's flowing around that actually cushions our nervous system. So that way when we do herky-jerky movements, we're not hurting our brain that's floating around in there. And it's also at a consistent pressure. And we'll talk about why that's important in a minute. However, even though it's there to act like shock absorbers and so our brain doesn't damage itself inside its bone case of a skull, unfortunately, violent things like whiplash and taking hard hits to the head and getting concussions over and over again can do a lot of damage. And we've discovered this definitely through over the years, especially looking at wrestlers like in WWE, or even before it was WWE and WWF, or whatever wrestling format you enjoy watching, if you enjoy watching it. And also come up several times in NFL players and football players. We really worry about them taking hard hits to the head, even with a helmet on. That doesn't stop the brain when taking a hard hit from going ka-chunk in the skull. And even with the cerebrospinal fluid to kind of keep it there and absorb shocks, it still can take a lot of damage, and that kills neurons, and that can change your brain chemistry and a lot of other things about your brain, which we're going to talk about in the next half of this chapter when we talk about the frontal lobe if it gets damaged, and our dear friend Phineas Gage. That's coming up in the next section. You'll get to learn all about poor Phineas Gage. And then, last but not least, is the pia mater, which is this thin, contains many nerves and blood vessels to nourish the brain and the spinal cord, hugs the brain following its bumps and contours. And again, those bumps and contours actually have names, which is actually really cool. So, and then fourth and last, that is not appearing in these notes, is toe mater, who is a character in Cars. And when I was rewriting these notes, my son thought that was very funny, and so I stuck it in there. And if you like the movie Cars, then there you go. Anyway, so again, here's the picture of it. So here's the dura mater right here. And it's got a periosteal layer and a meningeal layer. And again, right in here, we have this subarachnoid space. This is where the cerebrospinal fluid is flowing, and you'll also see it in another couple sections in a minute. Here's the arachnoid matter. It has all these little fingers coming down, and that reabsorbs the cerebrospinal fluid because we're constantly making it, putting it out, and reabsorbing it because we make more than we need, which is a good thing because it maintains a uniform pressure. And because we don't want too much pressure and we don't want too little pressure, because you don't want your brain just sloshing around like a wet thing in a jar. Again, damage. And if you don't have too much pressure, damage. We don't want damage to the brain. We like our brain undamaged, thank you. And then the pia mater, which is hugging basically the brain itself. And again, So, cerebral spinal fluid is formed in 4 interconnected cavities called the ventricles. So, we've got 4, but there's only 2 that are named after its numbers. So, we've got the lateral ventricles right here. We've got the interventricle foramen, which is kind of like, connecting them to the rest. You got the third ventricle right here, which is this part right here. And then we go down to the cerebral aqueduct, which goes down into here, which brings it to the 4th ventricle, which is looks small this way, but when you tear it frontal, it's bigger that way. And then, like the TARDIS, it's bigger on the inside. And then the central canal that actually goes down and dips around your spinal column and comes back up around the outside of your brain. And so that's basically how it flows. So the chyroid plexuses secrete the CSF. They're reddish, tiny, cauliflower-looking masses that specialized capillaries from the pia mater covered in specialized epidermal cells. So remember, that's one of the types of cells I talked about in the last chapter 10. Much like the blood barrier, this is a blood CFS barrier that blocks the passage of water-soluble substances into the brain. Why? Because it's kind of like picking up feedback from more than one sound source. And then you get that noise over the speakers and everybody covers their ears and screams. Because it's the same thing with hormones that we're putting out and stuff like that. There's a lot of things that we don't need our brain picking up because he is the originator of a lot of signals coming out of us. We do not need feedback coming in that way. We need to control the feedback that is coming into our brain so that way we're not sitting there and going, oh God, I just released hormones to tell my, testosterone to do something, but now it's telling, now my brain's confused because it's getting it too. So we need to control what the brain, the feed, the information coming into the brain so the brain can work at optimal conditions. So, cerebral spinal fluid is clear, somewhat viscous liquid. It has greater concentration of sodium and a lesser concentration of glucose and potassium than other fluids. That maintains a stable ionic concentration because remember, sodium outside, potassium inside. So we need more sodium up there in the brain to replace anybody that we're losing. Also lets the waste products leave the brain. So that's another reason is to remember what goes into cells still must come out even in the neurons in the brain. So we secrete about 500 milliliters of cerebral spinal fluid daily, but only about 140 milliliters are in the nervous system because it's continuously reabsorbed by those arachnoid granulations, those tiny finger-like structures that you saw back here. So it's constantly being reabsorbed. And test question. Right there. Yeah. Anyway, so now if you get too much, this is known as hydrocephaly, and we'll get into that in a minute, which is not great. So again, if you want to know how to flow, so here's where it's all created in these little red cauliflower parts right here. That's our friends, the chyroid plexuses, and there's also some down here too, next to the cerebellum. And so these guys make it, and it flows into all the ventricles, and then it goes down and down and down around the spine, and it comes up, and it goes up this way, as well as up and around this way, and around and around, and again we got another one going back in again. So it's constantly flowing around and removing wastes, preventing things, water soluble things from getting in, holding reserve of salt so that way you can maintain the constant differential in the nerves themselves. And that's why this is kind of, and it, keeps your brain in a stable environment so it's not just rattling around up there, because we don't need our brains rattling around more than they normally do. And this is, again, a, important part of maintaining your brain health. However, continuous secretion of reabsorption keeps the fluid pressure constant. Interference with this by infection, tumor, blood clot, damage can increase intracranial pressure, or ICP, in the ventricles, not to be confused with insane clown posse. This can lead to a collapse of cerebral blood vessels, injury of brain tissues, and remember, once a neuron dies, you cannot regenerate it, so the last thing we need is for damage to kill off our brain, because that's bad. We're not going to get that back. We can go through training to retrain our brains, which is actually really cool, and I'll talk about that in the next section. but we do not want too much pressure or too little pressure. A spinal tap or lumbar puncture is the only way we can measure CFS pressure. So yeah, we got to stick a needle in you. However, pressure can be relieved by insertion of a drain into the subarachnoid space. So if we're making too much, we can compensate that. And that's called hydrocephalus in an infant or water on the brain, which happens. Sometimes the baby is just making too much and we have to put a shunt in so that way it drains out so it doesn't hurt the developing brain. Too much pressure, bad. If you've ever heard pressure on the brain bad, this is why, because the cerebral spinal fluid. Because it can damage just as much of our brain going inside from, constant football hits or wrestling moves or whatever, because we're not protecting our brain enough. And there is a lot of science about that, and how can we reduce that while keeping our athletes healthy, because that does lead to major issues, including there was, and like I said, I'm going to go into this in a story with our dear Phineas Gage. You'll hear me in the next section talking about that, but there was a wrestler who unfortunately took too many hits, and it did change him, led to depression, and unfortunately also led him to commit a murder, unlifing himself. It was not a good thing, but a lot of people point back to that, going, it's because of all the hits he took to his head, because he was known to be a very hardcore wrestler and taking all the big hits to make it look really good for the television. So unfortunately that builds up over time. That level of brain damage builds up, and it can damage especially the frontal lobe, which is where a lot of things can lie. Also the limbic system, which we'll get into in the next group. So anyway, so speaking of brains, they age like the rest of us. So before we get into all the cool nitty-gritty stuff, let's go over how our brains age. Now, I mentioned here, eat your blueberries. It's actually shown that just the texture of blueberries alone actually kind of excites parts of our brain that normally don't get excited on an everyday level. So yeah, actually eating blueberries, the sensory sensation itself actually gives your brain a bit of an exercise. So it isn't just the fact that they're packed full of good nutrients for you, it's also the act of eating them themselves that's kind of like our brain goes woo to it. So it's kind of weird. So eat your blueberries. Brain cells begin to die before birth due to apoptosis, a form of normal programmed cell death, because we actually are born with way more than we need, which is a good thing. It is better to be born with more than you need than less. And I'll talk about the Zika virus and what that did to people's brains in the next lecture as well. That's just sad. So anyway, over our average lifetime, the brain shrinks 10%. And I'm sure there's times in your life where you have literally felt it shrinking. I know I have. Sometimes they were in faculty meetings. And I don't mean here at Blue Ridge. I meant when I was teaching high school. There was moments where I'm just like, Anyway, more gray matter than white matter is lost with aging. Many cells die in the temporal lobes, but very few in the brainstem. Interestingly enough, the brainstem, and we'll go over this in the next lecture as well, is actually one of the older parts of your brain, evolutionarily speaking. Monty has a lot of the parts of his brain that are found in our brainstem, because he doesn't really have to think about thinking too hard. That, and he also has a human that takes care of him. So anyway, by the age of 90, our frontal cortex has lost half its neurons. Yeesh. So a number of dendritic branches in the cerebral cortex decreases, so, decreased levels of neurotransmitters between the neurons, so they're not communicating as efficiently. Action potential's propagation rate declines by 5 to 10%, which is why we get fading memories, slowed responses and reflexes. So this leads to an increase of fainting and falling down, and changes in sleep patterns that result in fewer sleeping hours. Like, I was just talking to my grandmother this past weekend, she's in her late 90s, and she's having a lot of back issues. She's unfortunately grown a hump, and because of this, she was usually a back sleeper, and she just can't get comfortable anymore because of the hump. And And now side sleeping has led to bone fragments floating around in her hip, and it's been a hot mess. So I feel for her greatly. She's definitely one that hasn't lost too much of her brain, I swear, because she is still sharp as a tack and has no problems telling me exactly how it is. So, oh, thank you, Grandma. So there you go. He goes over it as well. The central nervous system, our brain, our stem, and all that fun jazz. And yeah, this one's short because I should have split this chapter up a little better, but I did not. I need to go back and fix that by next semester. But anyway, long story short, with cheese, is that a lot of the stuff we're going to be talking about, the next lecture is going to be quite a bit longer. So A, brace yourselves. And B, oh yeah, I've got so many weird stories coming up in this next half of this lecture. I hope you enjoy. Some of them are sad, some of them are weird. So, prepare yourself. With that said, I'll see you in the next half in just a moment. See ya. Bye.